I'm just going to welcome you and get started. Just a little bit of a plug for some of the programs that are going on at the Learning Center right now. Summer camps are actually open, so if people would like to do some hands-on experiments and you haven't visited us yet, that's an option. And then also, we're almost at the end of the year here as far as the school year. There is one more round of the Meet a Scientist, here it is, series. So today we're in April, and we've got one more speaker, Jeremy Cito, who's a bioinformatician from CUNY City Tech, who will be joining us as the last speaker for the series. So it's my pleasure to welcome Dr. Ullas Pedmale, and he is going to talk about sensation, or how plants sense their environment.
Thank you, and thank you, DNA Learning Center, for giving me this opportunity. This is wonderful, and I'm very excited to tell you how plants sense their environment, and also talk a little bit about what we do in my lab at Costa Harbor. So today I would basically want to tell you how plants interact their environment. You may see plants are all around us, but I think, or probably you may not realize how smart or how competitive they are. So I'm going to touch a little bit about some of the behaviors of the plants and how they really interact with their environment. So I always like to start saying that we need plants, but plants don't need us, right? So we cannot exist without plants, but plants will be okay if you're not around, right? Because we really depend on plants directly or indirectly for the oxygen we breathe, the food, the clothes, and so on. Now, plants sustain life on Earth, as I said earlier, because of photosynthesis. We all depend on photosynthesis. You might have studied in your classes or different subjects where light is used by the plants as a source of energy to fix energy for us. And one of the byproducts of this photosynthesis is oxygen that we breathe in. And as you can see, this remarkable effect of photosynthesis on capturing carbon dioxide in the atmosphere. What you see here, this is a NASA satellite image where yellow is the carbon dioxide concentration that peaks when the plants are not growing. That's in winter. And when this plant starts growing, especially if you focus more in the United States, Midwest, when all these crops and plants come back to life in spring and summer, you can start seeing the amount of carbon dioxide that gets reduced in the atmosphere. This is the remarkable feat that plant does. Now, we got 5 senses, right? We can feel, we have a sense of touch. And I would like to share that plants can also feel being touched. A good example is Venus flytrap that can sense when an insect has landed, and it can close its leaves so that it can start using the fly as a source of energy, right? It's going to start digesting this fly that it has trapped and use it as a source of energy. Another good example is Mimosa pudica, or touch-me-not plant, where the leaves or the leaflet starts closing as soon as somebody touches it, right? So plants can really feel that they're being touched. Another example I'd like to give you is plants can taste like us. This is a nice example that was recently published. This is Arabidopsis, and so it's grown on plates that contain no salt or with some salt, okay? NaCl is nothing but the common salt that we use in our kitchen, in our food. And you can see this, the roots, before they encounter the salt layer, they start turning away, right? They start going away from the salt layer, because definitely the roots, they do not, or the plants, they do not want the roots to get pickled. So they're trying to move away from salt and get away from nasty stuff, okay? So they can sense there's a gradient of salt present in growth. Another example I like to share is about plants can smell. This is a parasitic plant called Cuscuta or dodder, where, as you can see, it can completely, in this image, the daughter doesn't make any leaves because it's a parasite. It doesn't need to undergo any photosynthesis. So what it does, as you can see, it has completely engulfed this tree, right? And slowly it's going to suffocate and basically eat that plant. The way Cuscuta sniffs its prey, because many plants secrete volatile compounds. Okay, this is a nice example. This baby Cuscuta just germinated, and on the left side is a tomato plant, and on the right is a grass. Okay, now it prefers a tomato plant because tomatoes secrete a compound that Cuscuta likes. So basically, like a snake, sniffs its prey, right? It uses its tongue to identify volatile compounds in the air. The same way Cuscuta can sniff out its prey, which is a tomato plant. It can latch on and be a parasite on that plant. And another example I'd like to give you is how some seeds, for years, they stay dormant until it has been exposed to smoke from a fire. So an example is in 2013, there was a wildfire in Mount Diablo in California, Northern California. And in response to that, some plants, for example, golden eardrops that were not seen for 50 years, growing in that piece of land right here, can germinate. The reason the plants like to wait so long for a forest fire to come through, so that it, as you can see in this image, many of the trees have been burned. When the trees are not there, it gets full access to sunlight, okay? So some plants are willing to wait for a really, really long time till they smell fire. And scientists have figured out what is this compound. So in this smoke, there is a compound called catechin that the seeds, okay? So you may think the seeds are dormant, but actually it's sniffing, it's looking at its environment. And when it smells catechin in the smoke, then the seeds undergo germination because it knows now the canopy or the trees are no longer there. It has full access to all the resources, right? Above ground resources as well as below ground nutrients. So this is another form of example where plants use different senses. Another good example is gravity. So plants know where to put their roots. They always tend to put their roots below ground, right? And scientists have identified mutants where they're agravitropic. They do not know the sense of gravity, or they cannot feel, so that it, as you can see here, the roots are going all over the place. So before I tell about light environment, so my lab is mostly interested in light, I want to remind you that plants build and modify their body parts based on their environment. If you look outside, or if you're around plants and trees, no two trees or plants look alike. The reason is, unlike animals or me, I was born with 5 fingers on each hand because it's set in my when I was born, the plant is already set, but it's not for the plants. The plants can constantly make leaves and branches, right? That's why we mow the grass, and the grass keeps coming up, right? Because of the incredible ability of the stem cells that is present in the shoot as well as at the root that keep giving rise to different body parts. So this makes it extremely flexible. And now why this plasticity of flexibility in making body parts is important? Because geography of a plant is its destiny, right? So unlike you and me, if there is, let's say it's raining, we can come inside into a shelter, but the plants are unable to do that. They're stuck in one place. Wherever the seed germinates, they start as to grow there and complete their life cycle. This is a wonderful example of how plants adapt. In 1959, there was a volcano in Hawaii, and after the volcano dies, this is the crater, right? And the seeds are germinated of this tree. And look at this in 2016, this bunch of plants and this tree really growing in this lava-filled, once a lava-filled crater. So plants are masters of adaptation. Now, plants, you'll find plants in all forms of habitats. this is a barbecue area. As you can see, a seed that might have landed between the cracks of the bricks and the top. It has found refuge, has sprouted, and almost about to finish its life cycle. It has already flowers, and you'll find plants growing even in desert. There are a lot of factors that affect a plant's growth and development: light, its genetic makeup, water, carbon availability, temperature is a big factor, nutrients, and competition with pathogens and herbivores, including grazing animals, cows, goats, etc. in the wild. So what I'm going to tell you is the light environment of the plant. Now, as I've told you earlier in my first or second slide, how light provides energy to the plant for photosynthesis, but plants also use light as a source of information to tell them where they are on the planet. It helps the plant tell, hey, whether the spring has come or the fall is arriving, right? This is when we see a lot of deciduous trees and plants, they lose their leaves in preparation for winter to come. So all this information is fed by light environment. How does a plant know the winter is arriving or the spring is around the corner? The reason is during fall or in winter, the days are getting shorter and shorter. And now when spring approaches, the days are getting longer and longer. And this is all about the light information that plant gets. Now light plays a major role in growth and development. It helps the plant transition to make chloroplast, okay? The chloroplast is an organelle like mitochondria that's present in all organisms. That's a source of energy. Chloroplast contain chlorophyll, which helps in light capture and where the photosynthesis happens, right? So light helps the plant do this transition. And light can also influence growth. So this is a nice example where we have taken Arabidopsis seedlings. So Arabidopsis is one of the model systems that we use in our lab. And you can see in this example, as we increase the light intensity from 0, you can see the plants are skinny and there is no open leaves of cotyledon, right? Because if there is no light, it need not make a chloroplast. It doesn't want to spend its energy. It's long because it thinks it's deep in the ground and it's trying to grow to seek light. And this is the basis of sprouts. So if you eat any sprout, basically you germinate seeds without light and you get this long, slinky seedling. Now, as you start giving more and more light, you can see the hypocotyl or the seedling stem get shorter and shorter. So this is a great quantitative measurement for us in the lab, and we can ask a lot of questions based on this very simple readout, okay? Or basically you can see how light intensity influences stem growth. So this is a very reproducible phenomenon that we can observe in plants in response to light. Now you may ask how plants can tell how much light it has seen. Going back to my previous slide, you can see that with increase or changes in light intensity, it affects stem growth. So how do plants know how much light and what color of light they are seeing? Since as we can see color, right, we have rods and cones, it gives us color vision. The plants also have color vision. So instead of rods and cones, plants have a bunch of specialized photoreceptors, phytochromes, cyclopas, cryptochromes, phototropins, and UVB receptors. So phytochromes sense red light and far red light. Cyclopas sense blue. So is cryptochrome and phototropins. They all sense blue light. But cryptochrome is the only class of photoreceptors that sense light that are present in both plants and animals. And plants can also sense UVB. Okay, so UV radiation is important for, important for plants because they're stuck in a place, right? It's always there out there in the sun. We do use sunscreen, but the plants are unable to use sunscreen. But this receptor or the eye can tell the plant how much of UV it is seeing so that it can also turn on some natural or can make its own sunscreen and protect its DNA from radiation causing mutations. Okay, then what do we study in my lab? Okay, and what we're trying to achieve and solve. So before I started my lab, I got my training in University of Missouri, where I did my PhD on how plants bend towards light. Okay, that is phototropism. I studied that. And after that, I did my postdoctoral training at the Salk Institute in San Diego under Dr. Joanne Corey. After that, I got my own position at Cold Spring Harbor. So what motivates me and coming to work daily is plants are such cool organisms. As I said, they're masters of adaptation. And whatever impact or whatever we can improve, understanding what we can learn from plants has a profound impact because we all depend on plants, right? Every person on the planet has to eat to survive. So whatever I do, it has a potential, or my lab does, has a potential to touch, if not hundreds, billions of people, right? So, so continuing with this, so our main goal in my lab is to understand how does the environment affects growth and development of an organism. So one of the examples I would like to give you in relation to this is you can see this two plants, but these two plants are of the same age, same genetic maker, but they're grown in two different light conditions. One is unshaded and other one is shaded environment. As you can see, just two different light conditions, you can see how different, radically different they are. In an unshaded light, this Arabidopsis seedling is super happy. It has thick green, lot of leaves. It's making a lot of secondary lateral branches, and it has a long primary root, whereas the shaded plant, it's a little bit anemic. So what is shade avoidance? So shade avoidance is when, so all crops, they do not like shade. What is shade? Shade is when plants are getting shaded by the neighbors, by neighboring plants, and the plants can tell. And this is one of the biggest factor that is facing farmers and scientists like me. How can we start increasing planting density? Okay, so more planting density a farmer can use, the more crops we can grow in a given area. And this is extremely important because the population of the planet is growing, but the number of, or the area that we can grow crops is shrinking because as the population grows, you need more area to build houses, etc. And also because of the climate change, right? That we are seeing some areas that were good to grow some kind of crops, then won't be good anymore. So it's become important that we understand how plants respond to shade and how we can solve this. So this is really it's motivating for me. If we understand the problem, why plants respond and how they respond to shade, then maybe we can engineer plants that won't be responding to shade, okay? And this is a nice example. As you can see, this is tomato plants. As you start growing them in higher and higher density, you can see the plants in them, they start getting longer and longer, okay? As you can see, plants are quite smart. They are able to tell that the density is getting increasing. They tend to get taller. So why do they grow taller? Because they cannot run away. Their only option is to get taller so that you can capture more sunlight than your neighbors. That is the idea of growing taller. But now this competition, right? I told you, plants might look docile, but actually they're very, very competitive in nature. So why do they do this? So this rapid growth, okay, this is actually happens really fast. So when they get shaded, and the plants can tell a shade from a plant, from that to a cloud, or from a building, or from a human or an animal staying on next to it, right? So this competition, this rapid growth of the shoots, the stem, and the petioles or the leaf stalks come at a cost. As you can see, the plants that are grown in high density they're extremely unfit. Basically, when there is a heavy rain or heavy rain or rain or winds, they get lost. This is wheat, which are grown in two different densities. This is normal density, and when you grow them in high density, they're getting shaded by us because you're cramming in more plants. They're really unfit. Okay, so you basically lose a lot of crops. And another thing, you lose yield. This is a nice example in radish. Radish is a modified root. As you can see, a shaded radish, which is grown in high density, you can see you're going to get a smaller, root, or basically modified root. That is the radish that we eat, okay? Because you can see most of the energy is spent in growing taller. And look at this unshaded, which is grown in normal density. It has got access to light. It's making bigger radish or the crop, and the leaves are healthy and normal looking. And this is true in cereals, right? We depend on a lot of cereals for food: sorghum, maize, or corn, then wheat, rice. These are all monocots or belonging to the grass family. They also respond to shade. And look at the sorghum, which is shaded, as less biomass compared to the one that's unshaded. It has a lot of tillers or branches, right? So this, so shade has a profound effect on our food security. And plants are also smart. If they sense a direction from where it's getting shaded, right? It's going to stop putting leaves. So we understand what shade is. We can mimic that in the lab now. And basically, this is a change in ratio to far red light to red light. So far red light, human eyes cannot see, but the plants can easily sense it. So in the field or in the lab setting, we can give a directional shade source, in this case, far red. And you can see this is a maize plant, skips that region. It does not make any leaves here, I mean, compared to the control, because you get this normal distribution of phyllotaxy of the leaves. Now, what my lab is trying to understand is how do a shaded shoot, right, above ground shoot that can sense changes in light environment, in this case shade, whether it's getting shaded or not, how does it tell the root to grow or not to grow, okay? As you can see here, this is the common lab plant system, Arabidopsis. You can see the root is affected in shade, so is in tomato as well as in sorghum. In tomato and sorghum, there's hardly any root matter left. So this is an area we're trying to understand because we're also interested in long distance communications. As you know, plants do not have a brain or a central nervous system. So how does a shoot talk to the root, right? What is the nature of the long distance communication in plants? So we understand a lot how this stem grows in response to shade. There's a lot of work done in many labs, and there are low red to far red signals to low blue light signals that's coming from the sun. When the changes in this light quality, okay, and quantity changes, we understand the molecular mechanisms. For example, phytochrome, red, far red receptors are involved in this process. And in response to changes in blue light, cryptochromes 1 and 2 are present, and these invoke a series of signaling cascade that leads to the stem or hypocotyl growth. So my lab decided to understand more about this below ground problem, right? As you can understand, a poor root system affects the entire productivity of the plant because you need to have a good root system for having a healthy and productive plant. So to understand, I'm going to just briefly present you some results. We looked at the gene expression pattern in unshaded, and this is the WL means white light, unshaded and shaded plants. We did it in Arabidopsis and tomato. So we compared two species. So we just looked at the gene expression pattern in the dissected roots grown in these conditions in tomato and Arabidopsis, and we use this technique called RNA-seq. Basically, we can look at global gene expression patterns over time. So we did a time course. In case of Arabidopsis, we took samples at half an hour, 3 hours, so on until 5 days. And tomato, we started with 3 hours and stopped at 1 day. Okay, now what we found, so we found that in shade, the plants, based on the RNA-seq data, we found that the plants were turning on as if they've been attacked by a pathogen or they're under stress. So the gene expression pattern that a plant puts out when it's attacked by a bacteria or a fungi or a pathogenic fungi, I did the same thing. And we can visualize there are some markers that respond to such pathogens. And we can take these plants that express this marker, in this case, a blue color. So here, I'll just walk you with this example here. This is this marker plant. This is Arabidopsis. Whenever we mimic as if been infected by a bacteria. It's flagellin 22. It's made from the flagella of some pathogenic bacteria. So whenever the plant senses this flagellin 22, FLG22, it turns on this blue color. So for us, we can visualize where the plant is turning on a defense-like response, okay? Because of this, we can follow this blue color. Now we can take this plant, put them in unshaded, white light, or under shade. And as you can see, under shade, the roots, only the roots turn on this blue color, means the roots are, the shade is feeling that as if it's under attack by pathogens, or basically the same markers that turn on when the plants are getting stressed by pathogens or by drought, the same signatures are getting turned on. We can really visualize this in the root. Then postdoc in the lab, Dani Rosado, is originally from Brazil. She discovered that a large number of worky transcription factors, WRKY, in short, we call it worky transcription factors. They are specifically induced in the roots of shaded plants, both in Arabidopsis and tomato. And then she identified that if you always keep these transcription factors on, you can make the roots shorter and shorter. And what do these worky transcription factors do? They do come on when there is stress. And what is interesting that Dani found out is, is when you keep these genes or transcription factors on throughout, the plant roots do not grow much. As you can see, this is unshaded and shade. The plants, the roots are always shorter, right? So now we identified some factors that we can manipulate in crops that we can make a longer root. So this is an example I gave, and most of the work was done by my past and current lab members at Coachman Harvest. And some of the data I showed you was mostly done by Dani Rosado in the lab. And I'd like to thank the taxpayers or NIH and NSF for funding our work in the lab. Thank you for your attention. I would like to take questions from you.